We're going to talk about pathways and interactions. My goal is to just give an overview of the ways that you can use different approaches that take information that we already know about genes and gene networks and how genes relate to proteins and kind of inform your selection of SNPs or use the SNP data in a slightly different way to give yourself a little bit more power. So that's my goal. As I'm going, I prepared a lot less this time because people were talking so much last time. So if you don't talk, then it's going to be like 20 minutes. So ask questions for sure, jump in, and we'll go from there. So I'll start with gene-level analyses. So one of the big problems that we've already talked about, you're looking at 600 people, and your p-value, your alpha is 5 times 10 to the negative 8th. So one way that you can increase your power a little bit is to summarize some of those SNPs into a functional unit that we already know is meaningful. And actually, the unit that we think these SNPs are operating at is a gene level. So if you just run all the genes across the genome instead of all the SNPs, you have about 20,000 genes in the human genome. So your alpha, instead of being 5 times 10 to the negative 8, is now 2.5 times 10 to the negative 6. So hey, we just added, I just increased your statistical power and you can write a grant. So you don't need to come later. That's the story for your grant. The interpretation is also a lot easier at the gene level. So with SNPs, what you've already seen, and I'll talk about this a little bit later more, and how you can try to learn some information even when SNPs are in a hard location. But spoiler alert, when you run your GWAS, you're going to end up with SNPs that are in non-coding regions of the genome, usually intergenic, and you're going to have no idea what gene they map to. So if you use the gene level analysis, you kind of take care of that whole problem by focusing in on a functional unit that you sort of pretend, at least, that you understand. So it helps with interpretation. The non-coding issue is less of an issue, and it kind of increases power, assuming that the way that you summarize the gene is actually correct, right? So I'll show you what I mean by that. Yes?
So if the SNP is on a non-coding region, does that mean it definitely doesn't have any effect?
It doesn't mean that, no. But it means that you don't know what effect it has.
So you're just saying, let's not bother with those because there's no way we can know.
I'm actually not saying that.
Oh, okay.
No, not that. But I'll show you what I am saying. So what we can do in a gene level analysis is we can summarize these SNPs across genes. The problem is, how do you decide exactly what Teresa's asking? How do you decide what SNP belongs to what gene? So it's a huge challenge. There's a lot of different ways to do this. And I'll show you one example of a really simple way. And then I'm actually going to show you a tool that you can use online where you just take your GWAS data and you send it to really smart people, and they send you back gene level results. And then they probably steal your data and do something with it. I have no idea. But I don't do it, so it's not my problem. So some of the issues you kind of have to consider as you're doing a gene level analysis is, how do I compare a gene of 2 million bases to a gene that only has 6,000 bases if I'm using SNP data to evaluate that gene? And then how am I going to account for the covariance between SNPs? So there's a lot of tools that have already been developed that kind of tackle these three issues. And I'm going to show you some of the solutions, but there are better solutions as well. So the most common type of gene level analysis, yes?
Sorry to interrupt. What does gene size have to do with function?
It doesn't necessarily, well, it does, but that's the reason I'm bringing it up is a statistical problem, not a functional problem. So statistically, let's just say that what we wanted to do is, I'm going to say, all right, I'm going to evaluate whether or not this gene has an association. The way I'm going to define it is, I'm going to take the gene region, I'm going to add 50 kb to each side, which I'm about to tell you is what we're going to do in this analysis. Well, the problem is, if I now evaluate all those SNPs and I correct for the total number of SNPs, I have, in the first gene that I showed, I have 2,300 possibilities of SNPs. In that region, it's not going to be 2,300, or I'm sorry, 2 million, but it's a lot more SNPs, right? So now, if I just correct for the total number of analyses I ran, I put a bias into my model, because now the chance of having a PT-PRD association is higher just because your type 1 error on that gene is higher. Does that make sense? And now the gene where you have 6,000 bases is actually underpowered completely, because you're correcting for all these bases and other genes, and you're not taking into account that this is actually a small gene, you only have a couple variants. So it's a statistical problem, not a functional problem, if that makes sense. So this is the Plink set-based analysis. You can run this right in Plink. using the same software that you're using yesterday when you learned how to run a GWAS. Put in a couple different -- commands, and all of a sudden you've got a set-based analysis instead. So you can do this right now. So what Plink does is it first wants you to select a set of SNPs. So how are we going to define a set of SNPs? And usually what we do is we go to where the actual gene region is within the genome, and we say, I'm going to go up 50 kb and down 50 kb from that gene. And there's actually other numbers that you can use. Instead of 50 kb, some of the functional data has told us there are actually better numbers than you actually want to go a little bit further on the front and not as far on the back. But you can use this to define what your gene region is. So why would you want to go 50 kb up and down from the gene? Yes, exactly. Regulators, right? So when we were talking about this in day one, I mentioned that prior to the gene, if you're thinking about the way that gene expression takes place, the way that you go from DNA to RNA transcription is there's this coding area of the gene upstream of the gene that regulates how that gene is expressed. So you want to include that regulation area in your gene definition. So this is just the steps I'll go through, and then if you have questions at the end of it, I'll take those. So the first thing you do is you just run a univariate analysis of all your SNPs. You can do LD pruning, which we've talked about, to kind of get rid of SNPs that are the same. And then what Plink does is it selects up to 5 of the SNPs with the lowest p-value, and it assigns a score based on the average chi-square of those SNPs. I'm going to come back and explain this in more detail. You permute your phenotype, you calculate a permutation score, and then you correct for the total number of sets evaluated. So what you're doing is you're saying, I already have a genome-wide analysis that's where we have associations between SNPs and my phenotype of interest. This is saying, how can I summarize, how can I come up with a score that says, what's the gene association? So not what's the SNP association, but in this region, what's my overall association with my phenotype? And all this is doing is just using a really kind of cute statistical trick by saying, now I'm going to convert those p-values to a chi-squared statistic, for example, where now all of those are on the same scale, and I can just sum them together to create a score. And then I can say, is that score better or worse than expected just given a random phenotype, right? So now I permute my phenotypes, and I end up with an empirical p-value that tells me, is that score better than would be expected if I were to just randomly assign it? Does that make sense? And then the last step is just a correction for multiple comparisons like you would do anyway. So when you're running this in Plink, you decide what your sets are. You tell Plink, like, here are my sets that I want to look at. But usually if you're if you're doing an entire gene-based analysis, you'd give it the definition for all the genes in the genome. Does that make sense? So here, there's a couple other approaches to gene-level analysis that kind of build on this same premises. And you'll see these, especially with rare variant analyses, a lot of these gene-based analyses have become more popular because, again, we're operating at the level, the functional level of gene is helpful, but also because you have so few observations at some of these coding regions that you need to figure out a way to summarize across those regions to get a score for your gene. Does that make sense? So the same basic premise, you again are using weighting schemes to try to take into account different variants within the gene. The first example is called the burden test. So you'll see this, again, a lot of times in rare variant analyses. Instead of taking your p-value, so in the set-based statistic I was talking about, you're really operating at the level of your p-value. With the burden test, you're operating from your level of raw data. So you're going to summarize, you're going to say, all right, I only have, let's just say 5 SNPs in this gene, but I don't have a lot of observations at all these SNPs. So I'm going to assume that the minor allele at any of these SNPs could be associated with my phenotype in the same way. So instead of just evaluating one of them, now I'm going to take what's my homozygous minor allele count across these 5 SNPs and call that all one gene. Does that make sense? So the problem with doing that is your direction, right? Because the minor allele within a given gene isn't always associated with gene expression in the same direction. Like sometimes carrying the minor allele in this SNP is going to increase expression, and other times it's going to decrease expression. So this model is ignoring that problem and just saying, well, on average, I'm going to assume that they're all operating in the same direction. I'm going to give it a score, and if they are, you're going to have a lot of power, and if they aren't, you're kind of sapping yourself. Does that issue make sense? So SKAT is another approach to to summarizing across a gene that takes a slightly different approach that kind of takes care of that burden problem that I just mentioned, right? So instead, what SKAT does, I think I'm actually just going to use the mixed effects summary of it instead, because I think it's a little bit easier to think of. It's basically you just take all the SNPs within the gene that you're talking about, treat that as your random effect in a mixed effects model, and ask, is my random effect associated with my outcome? So in that case, you're not as concerned about the burden because you're not doing a sum. You're not adding all of those together. You're allowing the direction to be different across them and just using the association as a sort of a joint effect together to ask, is that associated with my phenotype? So SKAT-O is a combination of those two things. So it uses the burden test and actually is just literally a linear combination of these two things. But it uses the burden test because there are occasions where the burden test is more powerful than SKAT-O when the variants are operating in the same direction and in the same way. And then it uses SKAT to kind of combine across the entire gene. So it kind of is the best of both worlds. So usually when you see this type of approach, you're going to see that they used SKAT-O. And that's what SKAT-O is. So you guys are now experts on rare variant analyses as well, and you can do that. Is there any questions on that? I know I flew through it. Yes?
So I remember a couple of papers say never the acknowledgements, always just the evaluations. So do you normally just get that SNP association for a gene region? You don't actually get
A beta. Yeah, it depends on which way you're doing it. So technically, if you did it the second way I said, you could calculate a beta. But for SKAT-O, you really don't have a true beta association because you're using the level of analysis that you're taking the summary at is at the summary statistic level. So, although Q, is there a way to, you would, is there a way to calculate a beta?
Yeah, well, that makes sense. So you can't really tell if the gene overall is having a positive effect or a negative effect.
There's a value in SKAT-O from 0 to 1.
Yes.
It tells you whether it's more a burden test or it's giving you a p-value. So you get some idea of whether it's coming from one or the other.
Right, yeah. So there's a test statistic that comes out in SKAT-O that'll tell you where's the weighting coming from. So it's not the equivalent to a beta coefficient like these SNPs are associated, for a 1 point increase in your summary of your SNPs, it's associated with a 1 standard deviation increase in your outcome or something like that. But you can get a test statistic that tells you where is the weighting coming from. Is this more of a burden test association or is this more of a SKAT association? So Vegas is basically set-based analysis applied directly to your GWAS p-values. So this is a really creative way of using your GWAS test statistics and being able to evaluate at the gene level. So what it does is it converts your p-value to a chi-squared statistic in a 1 degree of freedom chi-square test, and then it uses that chi-square to calculate the gene level statistic, just like what the set-based analysis does in Plink. And then adjusts for LD using 1,000 genomes, and it runs simulations to calculate your empirical p-value. So just the same idea as doing the permutation, but it uses a simulation approach instead. So this analysis, because it's operating at the level of your GWAS test, your GWAS summary statistics, it can be applied to any data set where you've run a GWAS, and there's a tool called Vegas 2 that will actually run this for you. So you can take your results from yesterday. I actually should have done this, and I didn't, but you could just take your results from yesterday, go onto this web page, upload it, and the next day you'll get an email back that says, here's your gene level associations. Yep.
So I just want to say this summary sentence so I can get the whole point of what's going on.
Yes.
So basically, instead of looking at individual SNPs and seeing which ones are important, you're using all that. You're instead trying to find which genes have the most importance based off of, the SNP information within the gene.
Exactly.
That's all. I just wanted to make sure.
Yeah.
And just to take that even more to a more ignorant level, the SNPs are very small components of a gene. So multiple SNPs can underlie one gene. There's no overlap, so there's no SNP that can have 2 genes associated with
Yeah, so there is. Yeah.
So you don't necessarily know. It's a bit of a hit in the dark, but then you're like, well, it could be from a couple of different genes.
Yeah, so it's a big problem when you get into some of the regions that we saw two days ago, or was it whatever? It's a big problem when you have a lot of genes that are very close together, right? Because you don't know the SNP may be 50 kb up and downstream from a whole bunch of genes, and there's not a right pick.
So even filtering on that LD doesn't...
It's not enough, yeah. You can't, because you can't differentiate what's a SNP actually doing. The only way to do it is to actually go into cells, modify that SNP, and say what's the association with the gene.
Some genes are embedded in the introns of other genes. Oh, so in those cases, there's no way to see.
So the point about the summary statistics versus the GWAS data themselves is an important one to think about. So we have person-level SNP data from ADNI that you ran. So person number 4 had which variants at which place, right, for 500,000 data points. That's a very large data set, and it's incredibly identified. In the state of Washington at one point, the law was that if you had 10 SNPs, it was identified. And we have 500,000 SNPs, right? So it's absolutely a unique marker of an individual person. Not that you can walk down the street and say, I know what your SNPs are, right? You need to have a blood sample and the person's eye. So, but still, there's some personal privacy concerns, especially that studies and IRBs have for releasing individual level genotype data. However, things like IGAP, when you have a meta-analysis, there's two issues. One is it's much more compact because at each locus you have an effect size and a p-value full stop period. That's it. Whereas with ADNI data, even though it's only 700 people or whatever we had, that's a whole lot more data per SNP because you need 700 individual level observations. So the summary data is smashed down, so it's one value per, and they're much more available. We saw a website a couple days ago from the Broad that says, hey, whatever your phenotype, download your summary data here and we'll share it with the world. And I can tell you PIs of studies that have provided me with individual level data are okay with me publishing and making available to the world summary data for my subgroups. So it's a big, important privacy practical issue of what's the starting data for this technology. And for SKAT, you need individual level genotype data. For Vegas, you don't. You can start with p-values that are much more readily available as a secondary data consumer. It's a lot easier to do this.
And there are an increasing number of programs
that
will take summary statistics as input rather than the
tons.
genotype data.
Yeah, I'll talk about another one in just a second. Yep.
A question related to what Rachel said. So if, as we increase the number of SNPs we have less clarity as to where they may be sitting, or higher probabilities that we can't necessarily identify what gene the SNP might be in. Does that tell us anything in terms of how many summary statistics to include? I mean, is it always better to just throw the whole thing in? Or?
No, because it's not really number; it's really location that matters. You know, so. Yeah, I mean, you could go through and say, like, how confident am I in so-and-so gene? But I'll show you another method to integrate gene-level data that I think is better because I use it. So there's a project that was developed a few years ago called GTech. And the idea was to try to get at this problem that we're talking about of annotation. Like, how do I know what SNP is regulating what gene and what tissue? And the problem is tissue expression is different for a given gene SNP association. So just if you were to look in the brain and say, is my SNP associated with prefrontal cortex of the gene right next to it, the answer that you get is not the same as if you look in the heart, right? And intuitively that makes sense because genes do different things in different tissues. The proteins that you need are different in different tissues. So it's not surprising that that's the case, but we don't always think about it that way. So what GTech did is it had a whole group of individuals who donated all 44 different tissues from the same individual. Everyone donated all 44 of those tissues. So now for a given individual, you have the association between a DNA SNP and tissue gene expression in 44 different tissues. Does that make sense? Yeah.
How parcellated was the brain? Was it like the brain, or was it frontal lobe, cingulate?
Yeah, I think there was 10 brain regions. It wasn't super parcellated. There's better brain projects going on now. The developer of this group, one of the developers is at Vanderbilt, and whenever she talks about this, she'll always say, the biggest differences are not between brain regions, it's between brain and everything else. I don't know that I completely agree with that, but I think there are the biggest differences that you're going to see are absolutely going to be at that level. But we have to look very carefully between regions too, because there are regions, just because the size of the difference between those two is larger doesn't mean that there's not absolutely essential differences between brain regions. Does that make sense? Yeah.
Whenever tissue level differences, does it have more to do with that gene is just not being expressed in that tissue versus it's not being regulated?
Yeah, you can't.
Like an on or off thing.
Yeah, so it's more, and if the eQTL association is different, it's most likely more that the SNP association is different, because if it's not expressed in that tissue, you won't have a statistic, right? Does that make sense? So, okay, so you have this GTech database. You can go online, gtechportal.com. You can go on there and you can take your SNP that you found yesterday, throw it up on GTech and say, is it associated with expression in any tissue? So the other way that you can use this, that's what I just said, yeah, right, is my SNP associated with gene expression in, so let's say, brain tissue? So one thing I'll say, so in GTech, they only have 125 individuals with brain data. So the sample size for these associations is very, very small, and there's a lot of projects ongoing now to try to increase the sample that we have. I'm also going to tell you about a few other resources. There's a resource called the AMP-AD project, which specifically looks at prefrontal cortex expression in multiple data sets across the US, and that has closer to 1,000 individuals with prefrontal cortex expression, if you summarize across those. So there are other resources that you can use to get these associations as well. Yeah.
I was going to say Brainiac has a...
And Brainiac, yep. And I'll have a link.
Which is easy.
Yes, yep. And I'll have a link for Brainiac on here too that you can use. Yes?
Why is more better in this case?
Yeah, okay. So I'll show you why. I'll come back to that in just a second. Yes?
Is the SNP gene expression different and like different?
Are eQTL associations different across races? I don't know. Probably.
Intuitively, they have to be
Right.
different SNPs.
Yes, right.
And then you're in the sample probably mostly white.
Yes.
Yes. Almost exclusive.
You need a very diverse array. Like the good resource would have lots of people with lots of diversity.
Yeah. I think another thing to think about is that the GTEx data in general is from tissue homogeneity. So it's not a pure cell population, and obviously where you take it from in the brain is going to have different amounts of neurons versus glia. And if you compare that in AMP-AD data set, you have Alzheimer brains versus control brains.
Yep.
You're going to have completely different numbers of neurons in the same mass of brain tissue from an Alzheimer patient compared to a control. So you have to be wary of that. When you see something significant in that context, you need to be able to try and correct somehow for the differences in some way.
Yeah, whenever you think about expression, your first question should always be, what tissue is this coming from? And then your second question should be, what cell? And here we don't know the cell type, right? Because it's a conglomeration of all the cells that are in the brain. So if only there was a group working on cell sorting to do this kind of stuff, that would be what Allison's group's doing.
It's like, since we are looking at it seems like this is a huge problem for the interpretation of this data, given that we're interested in genetic variants associated with certain diseases.
I couldn't agree more. I won't use the brain data because I think it's very difficult to interpret. In fact, for all the GWAS genes I've looked at in AD, there's almost no eQTLs in brain tissue.
That's right.
And what we found is we could detect them in myeloid cells because it's probably picking up microglial expression, and you can't do that in a brain homogenate because there aren't enough microglia in there. It's a sensitivity problem. So yeah, I think as Tim said, we need some cell-specific data.
Yeah.
But it's going to take a little while before we have that.
Yep. And so this is really important because I'm about to tell you about a method that can use these data. And you might ask, well, why don't you just use brain eQTLs for it? And the reason we don't do that is because of what Allison just said, that the association between brain expression when it's calculated this way and AD isn't actually that good. It's not a great marker of expression differences between cases and controls. So because of that, you want to use kind of your power of this data, which is looking across other tissues that you may think aren't relevant, but it may tell you something about gene expression, gene regulation that is relevant to your disease. Does that make sense? So, okay, so let me tell you how we use it, and then you can ask questions about it. So yes, one way that you can use it is you can go in and say, is my SNP associated with gene expression? That alone, that question was not being asked when I started doing Alzheimer's disease research in 2012, right? How far we've come in thinking about regulation just to ask that question is pretty amazing and important. I think it's going to get a lot better. But the other thing that you can use with this data, which is really cool, is you can assume that gene expression is made up of these three components. You have factors driving your gene expression that you don't know. You have a component of your gene expression that's altered by your trait. And then you have this genetically regulated component. You have the SNP component, the SNP associations, the eQTLs that we were talking about, driving gene expression. And if you assume that, you can use those SNPs to impute your gene expression, and what you're then looking at is only the genetically regulated gene expression. Does that make sense? So you're isolating back to sort of causal association between SNP and expression, and then using that known knowledge to impute gene expression and use that as your predictor in your analysis. So in really, really simple terms, this is a gene-based analysis. That's all it is. But you're using what we know about real associations between SNPs and genes to use that SNP data in a better way. So now you're getting back to, you're taking care of a lot of the problems that we talked about where we don't know if this SNP is associated with expression. We don't know if it's regulating this gene. We're only looking at SNPs that we know are regulating a given gene in a given tissue, using that known association to build an expression trait that we can then use as our predictor in our model. Is everyone following that?
Do you know how many SNPs are associated with expression, say in brain? So that if you're multiple, how much are you decreasing your multiple test corrections?
Yes,
by using
I do know the
your
answer to this.
primary?
Yeah, so the big problem right now that I think Q actually has a paper under review right now, or going to be released soon on this topic. So what I just told you is you can't really go in and just take brain. You can do that. You can do that right now. You can go in, build these PrediXcan models in ADNI, only use brain expression and get an association. If you do that, you are really helping your multiple testing correction a lot. But if you look across all 44 tissues, now you've got all 20,000 genes, right? 44 tissues, you do the math, and all of a sudden your p-value correction is about 10 to the negative 7. So it hurts a little bit. So there's solutions that are being worked out now for how we can take care of that problem. And I do think there are ways that we can take care of it. Yep.
I need to go back to some basics into where the tissues are and view of the tissues are being taken. So the antibody is taking those tissues from cases.
And controls.
And controls.
Yeah.
Why do we need gene expression data from cases?
So the AMP-AD project is not designed to build PrediXcan models. The question is, are there differences in gene expression in the brain between cases and controls? That's why AMP-AD is happening. So, but we can use that resource to say, we probably don't want to use the AD cases at all, right? But we can use the controls to say, what's the association between a SNP and expression? And it's just a different way of using the data. So now we can use the knowledge that we've gained about eQTLs to inform our GWAS models. Does that make sense?
It might actually be, though, since AD brains are enriched for glia,
Yep.
if your genetic signal is coming from glia, you might have more power to look at each
The AD
from an
brains.
Alzheimer brain than from a control brain. I've not actually tried to do it.
I haven't either. I'll try that.
You're seeing that because the neurons are dying, yet not so much the
Yes.
Right.
on the same rate.
Yeah. So I would, one very sort of crude thing to say about MKD, which is millions of dollars, is that one of the initial analyses they did was demonstrate that there are more glia in an Alzheimer brain and there are more neurons in a control brain.
Great.
And so before we did the
Yep.
experiment.
Yep.
But Allison, the other thing that you're saying is that because there are more glia and that the expression is higher in glia, but not different then you may have better power to detect those tissue differences or regional differences.
Yes, you might have. I mean, obviously, one of the things also is that probably got a lot more activated microglia in an Alzheimer brain than you have in a control brain. So expression could be different for that reason, but I do think that you probably have more power to detect expression QTLs in an Alzheimer brain if they are microglia.
So I'm not going to show, I actually thought I put this in, but I didn't. You can actually calculate this from summary data as well. There's some really cool tricks that you can use about the association between a SNP and a trait to impute gene expression from the summary data, which is pretty cool. That's called PrediXcan instead of PrediXcan, but you can do that too. So I was asked to put up some resources so I just ran a GWAS and I found some SNPs. What do I do? Like, how do I find out this stuff that you're telling me that I don't know if it's regulating gene expression, I don't know where it is, I don't know if it's in a coding region or a non-coding region. So step one, if you go on to this dbSNP is a database from NCBI. You can go on there, you can put in your RS number, and it will show you a nice little map of here's where your SNP is, here's where all the genes that are in the region. You can find where the base pair location is, all that good stuff. On your SNP lookup table. If you want to know, is this an eQTL? Brainiac was mentioned. You can go on to Brainiac, put in your RS number. It'll tell you in all of the tissues in Brainiac, is there an association between that SNP and gene expression? What genes are associated with that SNP in the brain? If you want to look at regulation, there's this website called Haploreg, which will tell you about, is this SNP a regulator of gene expression? And a lot of that data is really likelihood data just given location and how much regulation happens in that location. But it can inform it a little bit. If you're wondering about LD between your SNP and another SNP, you can go on to this website. And this will all be, you guys can get this from the presentation, but I just wanted to walk you through each and figure out what the LD is between those SNPs. You can look at the protein atlas to figure out like, okay, I think it might be this gene. Is this gene even expressed in the brain? Is the protein that this gene codes expressed in the brain? Protein Atlas is a really good resource for that. And then there's all of these other tools that you can use on the Sanger site. You can go on there and there's all kinds of online tools that you can use to help inform your annotation and try to figure out what your SNP's doing. So it used to be that you'd run a GWAS and you'd have a hit and you'd say the nearest gene is X and you're done. And you can't do that now. Like, no reviewer is letting that through anymore. So now you have to make some statement about what's this SNP likely to be doing, is it likely to be regulatory, and what's my evidence that it's likely to be regulatory. So this is an expectation at this point if you're going to write up your GWAS finding from yesterday. So the finding yesterday was APOE, so don't write it up. Okay, so I just told you ways that we can analyze at the gene level. I'm going to walk very quickly through another thing that you can do called pathway analysis. So now what we're going to do is we're going to use known biological relationships between genes to try to figure out what pathways or what gene pathways might be associated with my phenotype of interest. So there's this resource called KEGG, which has a list of gene pathways. So you can kind of think of them as gene networks. And it makes assumptions about certain genes being involved in certain diseases but it's very well annotated and it's really easy to use. So a lot of times this is where we'll kind of start for our pathway analyses is known gene networks. So here's the example of the Alzheimer's disease network. So if I said that I want to know if the Alzheimer's network is associated with my disease, these are all the genes that I would be evaluating. So there's an analysis approach called pathway enrichment. And what you're asking is, given the associations that I have in my analysis, are there certain pathways that are showing up more than you would expect by chance? So the way that we do it is we define genes like we've already mentioned before. We get a gene score like we mentioned before. We decide what a high score is. So we look at what are my gene scores across the entire genome for this. I'm going to call a certain level high. 5% is usually what we use, so the 95th percentile. And then what we ask is, does a given pathway have more genes in that high score category than you would expect? So you look how many genes are coming up in this pathway, and then I'm going to compare that to 10,000 randomly selected genes. And then I'm going to use that to calculate my p-value. And when you run that kind of analysis, you get a result that looks something like this, that says like, oh yeah, you've got these different pathways that are enriched in your analysis, and you can make some statement about, in Alzheimer's disease, the immune pathway is showing up more than you'd expect. Does that approach make sense to everyone? Okay. And then I'm going to talk very quickly about gene interactions too, mostly to try to tell you not to ever do them. So statistical power. So we found out that statistical power in GWAS studies is a challenge, right? Our alpha is so incredibly low, and computationally it can take a little time. So how long did it take most people to run a GWAS yesterday? Like how long was your computer running? 30 seconds, a minute, something like that? Yeah. So now if we're talking about gene interactions, all of those things go through the roof. So now instead of 1 million SNPs, I'm going to use 1 million as the number for all of these things. The number could be slightly different. But now instead of 1 million tests you're running, you're running 1 million choose 2. Okay? So that's 499,999,500,000 tests. So your correction p-value is not 10 to the negative 8th anymore. It is now 1 times 10 to the negative 13th. So that's just a statistical problem, right? Because now my threshold for statistical significance is through the roof. Computationally, this is extremely challenging. So when I was assigned a gene-wide interaction analysis my first year working with ADGC, and I was like, how hard could this be? I'm going to throw it in my cluster. I'm going to give it 12 processors. I'm going to let it run in parallel, and I'm just going to let it go. So I had 12 processors running in parallel for 15 days, and it didn't complete the analysis. And then I was like, wow, that's really crazy. I can't believe it didn't work, and I hadn't sat down and done the math, and here's why, right? So I needed 578 days. So if I had given it a little more time, it would have completed, but I didn't, so instead, yeah, so computationally, it's a huge burden too. And then interpretation. So not only is it computationally very difficult to do, but even if you get a result, how in the world do you interpret this result? So in this case, if I'm a homozygous carrier of this SNP that I'm annotating to this gene, or I'm a homozygous carrier of this SNP annotated to this gene, my effect completely flips. So this analysis was actually a candidate gene analysis, and actually kind of makes sense. So GSK3 beta we know regulates the gamma secretase cleavage of APP, and we also know that it drives a pro-inflammatory response in microglial phagocytosis, so the clearance of amyloid. So in that case, it's really great. If you have high GSK3 expression, it's doing what it's supposed to do, and your amyloid levels are going to be lower, and that's what we're seeing. But in the case of APBB2, which codes the FE65 protein, which is known to bind to the amyloid intracellular domain, which we know only when that protein binds to that domain does it drive APP expression and GSK3 expression. And when that happens, it's now amplified, and now it's really, really bad because you have more amyloid. So I came in with a hypothesis that I knew, and still at the end, the story was so incredibly complicated that I've never gotten to the end of that story and someone's like, yeah, really cool, right? Because it's so complicated. So when you have a gene interaction analysis where you're going blindly into it and you end up with 2 SNP associations where you don't even know what those SNPs are annotating, it is incredibly challenging. You don't know if the homozygous minor carrier is associated with higher or lower expression. It could be either. And you don't know it for the other SNP either. You actually don't even know if they're coding anything, right? So anyway, so don't do it. So if you are going to do it, there's some ways that you can get around some of these problems. One is to do a candidate gene approach. I just gave you one example of that. In that case, our hypothesis was that genes that regulate tau may also regulate amyloidosis based on someone else's paper in a model system. In this case, we just said we're going to take the top 10 genes that are associated from the IGAP. And at the time, this was actually a pre-IGAP paper, so there were only 10, not 21. And we looked at the associations among those genes and found a couple associations. You can also use an informed pathway approach instead. So you can use some of those KEGG pathways that I mentioned before. And you can say, I'm only going to look at in a given pathway. So that's going to reduce your multiple testing correction, maybe down to a GWAS level. Or you can actually, one of the things that Paul mentioned is there's all these other resources that I've already mentioned today. We have annotation resources and regulation resources. We have things that we know about protein-protein interaction networks, and Joey's done some really cool stuff with that information. And you can take that and say, now I'm going to build a likelihood interaction model, and I'm only going to look at SNPs that are likely regulators, likely in regions that I care about, in pathways that I care about, and they're in a high probability to interact. And then I'll only evaluate those ones. So we did that in ADGC and found a couple of genes that seemed to be associated. Yeah, and I guess I didn't do the other approach that you could use. So yeah, so you can kind of use all this biological information to help reduce the statistical problem and the computational problem, but really without doing one of those things, if you want to look at interactions, it's going to be really difficult in a small sample size. Yes?
So one of the papers I was looking at last year or something, it's supposed to look at that here as a SNP interaction, not gene-gene. So they're proposing to do what you said at the start, aggregate all these SNPs into a gene region, and look at interactions between gene regions.
Yep. So that is actually what we did in that paper. Yeah. Mm-hmm. Yep.
So, and that worked out.
What's that?
So that got you some interesting results.
Yeah, I mean, I still think it's extremely challenging to interpret because even in those cases where you're summarizing across the gene, you still don't really know the association between those SNPs in that gene. So we are trying to use PrediXcan to do this now too, and I think that helps a little bit. But again, the multiple comparison problem is a really big one. So yeah, it's challenging for sure.
But Tim, do you think, are there any convincing ACOE other gene interactions? And so the factor we're most sure about, the variants we're most sure about.
Yep.
Do we know
yeah. So
anything that's really interactive?
So the very first analysis we ran before we ran this was that we just said we're going to look in ADGC, run APOE interactions with SNPs, and nothing. Really didn't find anything at all. And I think Young, when she did her stratified analysis, same thing, right? They saw a couple pretty close associations, but not much. And in that case, you're only looking at one, right? It's a GWAS correction at that point, because you're looking at everything interacting with APOE, and still there wasn't anything there. So it doesn't seem to be, at least what we've seen, is that there's not major SNP regulators of the APOE effect.
What about CETP? There's been some genetic analysis because they're both involved in cholesterol pathways.
Sure. I mean, it could be. But the effects are explaining an extremely small amount of variance, whatever it could be. So, yeah. I'm sure there are, but they're small.
Well, that was